Hi, my name is Akira, and today I'll be explaining protein overexpression, and we will also hear from Rochester iGEM about a unique example of how the synthetic biology technique can actually be used. Synthetic biology is really a wide-ranging field encompassing a variety of biological methods, and the concept of gene editing often seems futuristic and almost sci-fi, but it's actually a commonly used synthetic biology technique which can manipulate gene expression in a variety of situations. Today, we'll specifically be discussing protein overexpression in bacterial cells, and Rochester iGEM will talk about how they use this technique in photosynthetic cyanobacteria to make organisms which can remove carbon dioxide from the environment. In addition to its use of protein overexpression, Rochester iGEM's project also highlights how synbio can have important applications outside of just human health. So what is protein overexpression? In short, it is a method to produce larger amounts of a particular protein within a cell, which can change that cell's function. For more detail, let's hear from some members of Rochester iGEM who have actually put this method to use.
For bacteria like E. coli, for instance, you have your gene of interest in just a plasmid that you have marked by a selection marker, controlled by a promoter, and ideally it would also have a recognition tag at the end of it as well. So after successful transformation of your bacteria of choice, we'll say it's E. coli, from there, well, at least depending on how you screen, if you have a reporter gene attached to it as well, you can see if those, first off, if those E. coli have taken up your plasmid with your gene of interest in it. And then you can do some cloning you can do some like diagnostic techniques to ensure that your gene of interest is inside the bacteria, like send it for sequencing, do colony PCR, restriction digest analysis. And then after that confirmation, your bacteria can start to overexpress this gene of interest and can start to make it depending on how strong your promoter that you have in front of it is. So if you have a tag at the end of your protein of interest, what you can do is perform like a protein extraction and a protein purification by using the tag that you have attached to it to isolate just your protein of interest.
Thank you to Rochester iGEM for that great explanation. To overexpress a protein in bacteria, a plasmid, which is a circular piece of DNA found exclusively in prokaryotic cells with a specific set of sequences, is injected into cells. These sequences include the sequence coding for the protein of interest, a strong promoter upstream of the coding sequence to increase transcription of that gene, an origin of replication to allow for the replication of the plasmid when the bacterial cell divides and replicates, and sometimes restriction sites which allow for the plasmid DNA to be cut and pasted into other plasmids or DNA strands. Once your plasmid with the components you're interested in has been created, it must be transfected or moved into cells. This can be done in a few ways, including electroporation, as seen here, which uses an electrical current to allow the plasmid to pass through the outer membrane of the cell, or by putting the plasmid into a virus, which can then move into the cell quickly. Rochester iGEM is going to explain their method and how they got their plasmid into their cells.
The most common way to do it is homologous recombination. So cyanobacterial genomes have a lot of like neutral sites in there. So depending on which strain, there's a library of different neutral sites that you can recombine genes of interest into. However, that takes a lot of time. It's very time intensive and you have to do a lot of screening and you have to do it for a very long time to make sure that each and every copy of the chromosome has that recombinant in it. So we chose against that route. The other route that you could go, the one that we're opting for, is what's known as a shuttle vector. So shuttle vectors are plasmids that are specifically designed that have an origin of replication that specifically works in multiple different like species of bacteria. So the one that we have is an RSF1010 based plasmid that replicates in E. coli and in cyanobacteria. Albeit it has a low copy number because of this like ability to replicate in different like strains and species of bacteria, but it can be replicated in both E. coli and in cyanobacteria. So that is the method that we're using.
Great. Though transfection is a generally reliable technique, it is by no means a perfect technique, and researchers still must confirm that the plasmid has successfully been incorporated and is being expressed within the cell. Two methods to confirm expression of the plasmid are with fluorescent reporters, as seen here on the left, or by the addition of an antibiotic resistance gene. on this right, both of which are further additions to the plasmid, which must be added before transfection. With fluorescent reporters, the DNA sequence coding for a fluorescent protein such as GFP is added to the plasmid. If the plasmid DNA is successfully being expressed within the cell, that protein will light up at its specific wavelength, and microscopy can be used, as you can see here, to visualize the quantity and location for cells for which expression is successfully occurring. Adding an antibiotic resistance gene, on the other hand, utilizes a similar concept, but rather than visualizing cells expressing the transfected plasmid, researchers can select just those cells. If the plasmid has a gene conferring resistance to a particular antibiotic, cells which do not have that gene will die when treated with that particular antibiotic, leaving only the cells which correctly express your plasmid. As you can see on this bottom right, the resistant cells will continue to divide and become the majority of the population. Rochester iGEM is now going to explain how they did their selection and give a more specific explanation for why they did so.
We kind of makeshift made our own multiple cloning site, if you will, as there is a GFP marker within our backbone. So if we can use restriction enzymes to cut out that GFP and then replace it with our gene of interest, if we know that first off the recombination and the transformation and all the ligation steps worked, if we can screen the bacteria and say no, it does not glow green, because the absence of the green and the presence of like a colony that's growing tells us that A, there is no GFP there, and B, there is a successfully like closed plasmid that is successful in the first place.
Great! So once the plasmid is created with all of its necessary parts, transfected into your cells of interest, and expression has been confirmed, and expressing cells have been selected, overexpressing cells have now been successfully created. These novel, genetically modified cells and their increased quantity of a specific protein can be very useful, as proteins serve a variety of functions in cells, and a higher concentration of a protein can promote certain cellular activities and inhibit other ones. Rochester iGEM overexpressed the alpha and beta subunits of cytochrome B559, a protein involved in photosynthesis, the process in which carbon dioxide and water are converted into glucose and oxygen using energy from sunlight. With global carbon dioxide emissions being a major problem, solutions to remove carbon dioxide from the atmosphere are urgently needed, and Rochester iGEM is developing a biological, cell-based method to do so using cyanobacteria, which are a particular group of bacteria who can already perform photosynthesis on their own. Rochester iGEM is striving to improve the efficiency of this process using protein overexpression, and here's their reasoning as to why they specifically chose to overexpress the alpha and beta subunits of cytochrome B559.
The light that the cyanobacteria harvest makes electrons through photosynthesis, and those will go through and create electricity. So our goal is to optimize this process and produce more electricity. And these cytochromes are a part of photosynthesis in photosystem 2. And their function isn't really well characterized, but as far as our research has told us, they predict that there are some effects of reducing photoinhibition. So hopefully our cyanobacteria will be able to live longer in this electricity producing phase, thus producing more electricity. But again, since these proteins aren't super well characterized, we're not 100% sure what the effect is going to be, but the hope is that it increases electricity output.
In addition to this removal of carbon dioxide, Rochester iGEM also intends to redirect electrons produced in photosynthesis to create an electrical output and possibly even produce ethanol to produce energy and biological materials in addition to environmental remediation. Eventually, Rochester iGEM hopes to print these bacteria on a paper-like surface, creating a biological solar panel which uses the power of sunlight and genetically modified bacteria to address our global climate crisis. This is truly a multifaceted and impactful concept rooted in protein overexpression, and here's this team's thoughts on their ideal outcome for this project.
At the end of the day, we want this to be to show the world that there is a way to, get energy and have sustainable electricity without needing to put more CO2 back into our atmosphere. So take this carbon dioxide and put it get it out of our atmosphere while still being able to provide us with electricity that we can use. So we can envision this in like a power plant kind of system, where if you have like a big enough like a power plant that's just powered by cyanobacteria, we can have all of these panels that are hooked up to each other, and all of our cyanobacteria like hooked up in series and in parallel with each other, that can charge batteries, or it can power houses, or it can provide like electricity for like all sorts of uses. So ultimately, that's kind of what we see.
Thank you so much for that explanation. With proteins being such an important component of cellular function, modifying cells to overexpress particular proteins can have large-scale impacts and create cells which serve unique purposes. Rochester iGEM's project highlights just one example of how protein overexpression can be used to improve the world, and emphasizes how synbio can have crucial uses outside of human healthcare. In addition to global carbon emissions, many other problems arising around the world can be tackled with synbio, and iGEM as a whole serves to amplify this through its wide array of villages, including climate crisis, fashion and cosmetics, and even space. Synthetic biology has the potential to solve issues both within the human body and beyond it, and protein overexpression is an impactful technique to support this goal. Thank you so much to Rochester iGEM for this collaboration and creating this video.